Hopefully you can see my screen there. And yeah, thank you for that nice introduction and organizing this symposium. It's a really fun group that you've put together. And I'm happy to tell you today about our work studying natural vision in the mouse. So when we think about studying vision, we often think about something like this, an eye exam. So sitting stationary and trying to discriminate geometric symbols on a 2D screen. Whereas in fact, we actually use our vision for much more interesting types of things. For example, tracking a fast moving object in order to drive a motor output to intercept it. We use our vision to navigate through the world. And to demonstrate this, I really like this video from Mary Hayhoe's lab. This is the head-centered view of a person just walking down a path. And you can see how complex this visual scene is. First of all, trying to find your destination, trying to find the path in this rich textured environment, but also how this scene is jumping around as the head and the eye moves as you're walking. And then we also use our visual system to detect hidden objects. For example, this camouflaged octopus that's hidden in a complex three-dimensional natural scene. So despite the richness of how we use our visual system, we typically study it in the lab in a situation that's much more like this eye exam. In this case, a head-fixed mouse looking at geometrical stimuli on a two-dimensional screen. And so what I'd like to do today is tell you about our recent efforts to move beyond these types of paradigms to studying vision in a context that's much more similar to how we use it in the natural world. Here's a brief overview of what I'll talk about today. The majority of the talk will be focused on prey capture, which is an ethological task that we developed for mice. I'll tell you about some of the things we've learned about how the mouse visual system functions, what mice can see, how they move their eyes to do that, as well as some of the neural circuits that mediate this behavior. And then towards the end, I'll tell you about some new approaches that we've been developing to be able to study movement and vision together. So before I get into prey capture, actually, I want to take a step back and tell you how we got into studying natural behavior in the mouse. As Eric mentioned, we actually started studying mouse visual cortex about 15 years ago when I was a postdoc in Michael Stryker's lab, with the goal of using the genetic tools that are available in mouse to be able to answer some of the long-standing questions about visual cortex that had arisen from cat and monkey. How do you get orientation, selectivity, and so on? But a big question back then was, can you study vision in the mouse? Which isn't such a bad question. People that aren't mice blind. In fact, they do have very low acuity vision. So an image like this will end up looking much more like this to the mouse. They have roughly the equivalent of 22,000 vision. That's largely limited by the size of their eye. They have fewer photoreceptors, fewer retinal ganglion cells. So essentially that's like fewer megapixels in the detector. But the big question at the time was what aspects of visual processing are going to be conserved in the mouse visual cortex? Which aspects are still there despite this low acuity regime that they operate in? And so we did recordings measuring a large number of properties that have been known, going back to Hubel and Weasel, things like orientation, selectivity, and so on. The one I want to focus on today is the spatial structure of receptive fields. And in particular, Hubel and Weasel had shown that neurons in cat and monkey V1 respond primarily to edges or bars of light. And then we wanted to see whether that would be true in the mouse as well. So the approach that we use for this is what's known as the spike-triggered average receptive field. The way we calculate this is by presenting this noise stimulus to the mouse. These recordings were in an anesthetized mouse. We present the stimulus on a monitor in front of them while we're recording with silicon probes. Each time a cell fires a spike, we take the image that was on the screen shortly preceding that spike, collect all of these together, average them, and we get what's known as the spike-triggered average receptive field. Essentially, it's the average stimulus that preceded the neuron's firing. In this case, for this neuron, this is a light bar with dark edges. So matching very closely to what Hubel and Weasel described almost 50 years ago in the primate. I should mention the spike-triggered average is a relatively simple way of characterizing receptive fields. It was kind of standard at the time. I'll come back at the end of the talk to talk about some more advanced methods, but you can essentially think of this as just getting out the average stimulus that caused a cell to fire. So we took our data from the mouse and compared this with some data from Dario Ringach in the monkey. Here I've color-coded the receptive field so that regions where the neuron was responding to dark are in blue and light are in red. And you can see that, for example, this cell is responding to an elongated dark spot. This is responding to an edge between light and dark. and this one is responding to a dark bar with light edges. So in comparing this to monkey, one thing I should note is that these the scale bar here is 20 degrees and the scale bar here is 1 degree. So the monkey is responding to much smaller stimuli, but you can see that they're roughly similar. You may think that I just kind of chose the right examples and use the MATLAB color scheme so that everything looks roughly the same. But we were actually able to quantitatively compare these. For the vision aficionados, we fit these to Gabor functions, calculated a couple of scale-free parameters, nx and ny, that describe the structure of the receptive field, and showed that these are mouse and monkey lie in the same region of that parameter space. I should mention these are scale-free parameters, so they're comparing the shape of the receptive field distinct from the size of the receptive field. So at the time, this was very exciting. On the one hand, it was interesting finding about how comparative visual systems, the fact that monkey and mice are extracting similar features from the visual scene, just at different length scales, probably says something about how visual systems are wired to the statistics of natural scenes and kind of scale-free parameters of natural scenes. It was also, of course, very encouraging for the use of the mouse to study visual cortex, that if they're responding to similar stimuli, now we can go in and see how those mechanisms are being implemented. And of course, that's been fruitful for many labs. But one thing that I kind of glossed over here is the fact that these two scale bars are different. And one thing I like to do is plot these on the same scale bar, and you can actually see how dramatically different, even though the structure is the same, the size of the receptive fields really is dramatically different. And so this raised the question for us of, what kind of behaviors can this support? What behaviors are you going to do with receptive fields that are 5, 10, or 15 degrees across, as opposed to our receptive fields that are on the order of 1 degree or less across? What can you do without high acuity vision? So that was one of the things that motivated us to start looking at natural behavior. Another is a finding that we also made back in the Stryker lab when we started recording from awake mice. So we had adopted this technique that had been recently developed by David Tank's lab to put a mouse on a spherical treadmill. Of course, many labs have adopted the Tank lab's technique now. But when we first, so this is the setup here. The mouse is on a treadmill. He's held in place by a metal head plate that's affixed to the skull. We can lower an electrode down into cortex. Over here, there would be a computer monitor presenting visual stimuli. As you can see, the mouse is relatively comfortable. We can record for several hours at a time. But one thing we noticed relatively quickly was that there was a big difference between when the animal was sitting still and moving. As you can see, he spontaneously shifts between sitting still and moving. Here, he's actually grooming. But we noticed pretty quickly noticed that there was a big difference between the animal when they were stationary versus moving. This is a plot of spike rasters for repeated presentations of the exact same grading stimulus. You can see that in this case, the neuron fires spikes on every trial, every time we present the stimulus. But if we color code the trials as to whether the animal was sitting still in red versus moving in blue, you can already see that the cell fires more spikes when the animal's moving. We can see this if we look at the peristimulus time histogram. When the animal's stationary versus moving, the firing rates are about twice as high when the animal's moving. And in fact, if we look at the orientation tuning curve, you can see this looks like roughly a multiplicative or gain modulation of the tuning curve. The neurons are responding to the same orientations, but just with roughly twice the response amplitude. So it's really like the mouse, the mouse V1 is just turning up the gain on its visual responses, turning up the volume when it starts moving. So in my lab at Oregon, we spent a while trying to track down some of the circuits for this. So we use an optogenetic approach to study a region down in the brainstem, the mesencephalic locomotor region. We showed that this brain region was capable of both inducing locomotion, as you can see up above here, but also that it sent a projection up to the basal forebrain that was capable of inducing the gain effect in visual cortex, even in the absence of locomotion. So this region, the MLR, seems to be linking together movement and the effects up in cortex. And then the Stryker lab and Yang Dan's lab then went on to complete this circuit up to cortex, showing that cholinergic inputs, particularly to VIP neurons in V1, seem to be increasing the gain. I should also mention that a number of other labs have found that, that the effect of locomotion on visual responses is even more rich. For example, it causes changes in spatial integration and contextual integration. Other groups, particularly the Churchland and Carandini lab, have shown that these movement signals are really very widespread across the whole cortex. And the Churchland lab has shown that, even in the context of a visual task, these movement signals are much larger than the sensory responses themselves. And of course, people have often also found similar, although interestingly distinct effects on the auditory system. But while these types of approaches are really good at figuring out the circuitry, how do these gain changes happen? A much bigger question is why? What is the computational goal that the brain is trying to achieve by turning up the gain? And one of the big limitations in addressing this is the fact that most of these studies are performed in head-fixed animals, similar to the setup I showed a minute ago. And of course, that head-fixed approach is very powerful for things like being able to record from a large number of neurons at a time. It's also very useful for being able to present controlled visual stimuli and have a behavior where an animal can give a response by licking or moving a lever. But this introduces huge confounds in trying to figure out, like I mentioned, what is the goal? What is the animal trying to do when it turns up the gain on its visual processing during movement? And there's a number of limitations that this induces. One, kind of the most obvious, is the constraint on movement. If you don't allow the animal's head to move, you don't actually know what the action was they were trying to perform. Were they trying to move their head left or right? Were they approaching something? Or perhaps they were trying to avoid the stimulus that you're presenting. So again, we can't figure out the computational goal if we don't even know what the behavior was that the animal was trying to do. Another limitation is these non-natural stimuli. So when we present stimuli on a computer monitor, even if we aren't presenting these very boring grading patches, even if we present a natural scene on a monitor, that's still very different. A 2D image on a screen is quite different than the three-dimensional environment that we can move through and interact with in the real world. Another limitation is these types of behavior. The two alternative force choice, for example, is very different than the types of animal behavior that an animal actually performs in the wild. Many of you probably know that when you train an animal on this type of orientation discrimination, it can take the mouse 2 or 3 weeks just to learn horizontal versus vertical. They sit there every day staring at the monitor, verticals on this side, all they have to do is run to that side and they'll get the reward. But it takes them 2 or 3 weeks to make that association, which suggests that this isn't something that the animal's brain is wired to do. They aren't usually thinking, in the wild, they never have to say vertical gradings are on the right, I'm gonna go that way. But I think more than that, it points out that even though you can form those associations, and therefore this is a really good way to study things like learning or the cognitive aspects of decision making, is not the way that information is necessarily naturally routed in the mouse's, how visual information is actually routed and used in the mouse brain. And together this kind of implies that we're missing many of the regularities of the real world that the brain evolved to deal with. And on the one hand, that may just mean that we get lower firing rates because we aren't using the optimal stimuli. It may mean that we have to train our mice longer because it's not a natural behavior. But I'd also suggest that maybe it means that we are missing a large fraction of what the visual system actually does because we're just probing this region of parameter space that's very far from where it's actually evolved to perform. So this is what led us to look for natural behaviors in the mouse. So what are some of the mouse's natural behaviors? Well, one is fear responses. If a stimulus, a dark stimulus either looms or moves overhead, an animal will freeze or flee. And a number of groups have studied that behavior. Another is navigation, that the mouse has to use, in the wild, has to use its vision to find its way home and recognize landmarks. I also like to point out that one of the most kind of classic behavioral tasks for mice, the Morris water maze, is also inherently, even though it's usually used to study learning and memory, is inherently a vision task because the mouse has to learn where that platform is based on visual cues around the room. So mice clearly use their vision to perform navigation as well. But my postdoc, Jen Hoy, became interested in whether our lab mice would perform another natural behavior with vision, prey capture. She was inspired by this species. This is the grasshopper mouse. It's actually a very distantly related species of mouse to our lab mice. But it's often described as being specialized for our carnivorous lifestyle. These mice can catch crickets, scorpions, frogs. They have large claws and big teeth. And so people think of them as carnivores, as opposed to our lab mice, which it's essentially they sit in their cage and they eat the food pellets that we give them. But Jen was wondering whether our mice would actually catch crickets. And so she did the simple experiment of just dropping some crickets into the mouse's cage. Sure enough, the mice chase the crickets and they catch them. And so she was off and running and establishing this as a behavioral paradigm. So this is the setup that she developed. The mouse is in a white arena. We place a cricket in the arena with the mouse. I should mention that we do a little bit of habituation to the arena so the mice are comfortable and make sure that the mice are hungry. But once we do that, the mice will go straight towards the cricket. So you can probably see the mouse does these very long distance approaches directly towards the cricket's location. The mouse isn't quite as good at catching the cricket when it gets there. And this may be where some of those specializations for the carnivorous lifestyle come in. They don't have the big claws, they aren't quite as good at catching it. But what's clear is that they're able to detect the cricket at a distance and make a direct path towards the cricket's location. But of course, so, first of all, this was pretty exciting. Even our lab mice can still, despite being inbred and living in captivity their whole lives, they're still able to catch crickets. But the big question is, are they using vision to do this? So to test this, Jen did a series of sensory manipulations. So first, she deafened the animals by placing little earplugs in their ears. As we mentioned, we did a couple controls to make sure they really were deafened. But you can see that the mouse is still able to make these direct approaches towards the cricket's location. So they don't need hearing to be able to do that long-range targeting. On the other hand, if we place them in the dark, or actually under near infrared, so that we can film them, you'll see that the mouse pretty much doesn't realize that the cricket's there. Part of this is because the cricket really isn't making many auditory cues. They don't chirp in the presence of a predator, and the inherent sound they make is very minimal. Until you can see in this moment here, when the mouse bumps into the cricket, once the mouse knows where it is and he's actively chasing it, then he's able to, presumably use auditory cues to be able to continue pursuing the cricket and eventually catch it. And we think part of that is either that the mouse can use the auditory cues better when he's close up, or maybe just that the cricket is making more auditory cues once it's being actively pursued. Okay, so Jen quantified this. One of the measures that she used was how accurately the mouse aims towards the cricket's location, which we call the azimuth, mouse's head angle relative to where the cricket is. And you can see that when the animal is in the light, it's able to aim within about plus or minus 10 degrees towards the cricket's location. When we earplug them, that targeting is almost as accurate. Whereas once we put them in the dark, that targeting almost completely disappears. So there's still some very small bias towards the cricket's location from those moments when it happens to bump into it, but it's greatly reduced. And then once we earplug them and put them in the dark, they essentially never target the cricket, suggesting that other cues like olfaction aren't sufficient for this type of targeting. So Jen also generated this graph, which I think is a very powerful way of characterizing the mouse's visual system, visual capabilities. This is that same measure, how accurately are you aimed towards the cricket's location versus the distance to the cricket. First thing you can see is that when the animal's in the dark, these two curves up here, the animal never targets the cricket, never starts aiming towards the cricket. So they need vision to do that targeting. But then if we look at the shape of the curve, that you can see that starting around 15 to 20 centimeters away, the mouse starts aiming towards the cricket. So this tells us something about the distance over which the mouse visual system operates. Now I mentioned in the beginning, they have these relatively large receptive fields. And we used to think, well, that means you can either see large objects that are far away, or maybe small objects that are close up. But this shows that the mice can also operate in this intermediate range of 15 to 20 centimeters, identify objects in the environment in order to be able to approach them. Likewise, at 15 centimeters, when the mouse starts making these approaches, given the size of the cricket, about 1 centimeter, we can estimate the visual angle subtended by the cricket. So what's the size of stimuli that the mice can detect? And that ends up equating to about 5 degrees of visual angle, which turns out to be roughly the size of a retinal ganglion cell, a typical retinal ganglion cell receptive field. So this suggests that the mice are able to take the information at 5-degree receptive field and convert that into detecting a stimulus and driving behavior. And then finally, when the animal is actively pursuing these kind of close-up pursuits, they're able to aim to the cricket's location to within plus or minus 10 degrees. So this tells us something about the accuracy with which they can take that visual information and convert it into a motor output. So even though the cricket's trying to evade the mouse, they're still able to aim to within 10 degrees. So overall, this was nice. Even after 10 years of describing mouse highly selective receptive fields in the mouse, it was nice to see that they were able to take that visual information and convert it into a behavior, something that they do naturally, and that they seem to be using most of that information in the behavior. But also, I feel like a figure like this provides a way of kind of assessing the mouse's vision, but in the context of a natural task, to be able to quantify what the mouse can see, but in an ethological context, as opposed to in a trained artificial behavior. So this is a little bit about what mice can see. The next thing I want to address is how do they see that? In particular, how do mice move their eyes in order to be able to acquire information about a target? So of course, in humans, we survey the visual scene by using a series of saccades that allow us to place the fovea, our highest resolution vision, onto specific regions of the visual scene to acquire high resolution information. And then by scanning across, we can build up the representation of the scene. On the other hand, mice don't have a clear fovea. They do have a few regional specializations. They have UV cones that detect the upper visual field, and a few regions that have higher density, slightly higher density of retinal ganglion cells. However, they do have a binocular zone directly in front of them, about plus or minus, or a range of plus or minus 20 degrees, or 40 degrees total. And you can imagine that might be a useful area for them to target for things, to target their vision to for things that they're interested in. What had been known about eye movements in the rodent, and this was largely from a paper by Jason Kerr in rats, was that they do have compensatory eye movements. So much like us, if I move my head side to side, our eyes compensate to stabilize the visual scene. And Jason showed that rats do the same thing. It's a little bit interesting in rodents because their eyes are on the side of their head. So if you move this way, your eyes have to move together to compensate. Whereas if you tilt your head this way, the eyes have to move in opposite directions to compensate. So when you kind of put that together, you get what looks like very disconjugate movements, but still they're largely just compensating for the visual scene, the movement of your head to stabilize the visual scene, but with the added complication that your eyes are on the side of the head. However, obviously you can't always just compensate for head movements or you would never look at something new. So the question we want to address is where and how do mice target their gaze? How do they choose to direct their eyes to a new location? And of course, prey capture provides a perfect way to look at this because finally we have an idea of a distinct target that is salient to mouse and that's driving their visual behavior. So to figure out where the mice were looking, a really great grad student in the lab, Angie McHale, developed a system to implement a system for head-mounted eye cameras. So these are two little detachable cameras that hang out on arms, looking back at the mouse's eye. I should mention this is related to a system that had been published by Jasper Port's lab in 2018. And then actually concurrently, my next door neighbor here at University of Oregon, Mike Weir, published something similar to look at ear movements in the freely moving mouse for auditory processing. Using this head-mounted eye camera system, Angie was able to look at the mouse's eye movements as the mouse is pursuing and catching cricket. So you can see up here those eye movements. As I mentioned, they look highly chaotic. In actuality, these are very systematic. Most of what they're doing is compensating as the animal's head shifts. But again, it's a little interesting because their eyes are on the side of their head. You probably saw here, these two rays are showing the direction of the mouse's gaze, the left and the right eye. So the first thing that Angie wanted to look at was, how does movement affect eye movements? We already knew that most of the eye movements, or that a large fraction of the eye movements are compensatory, for, stabilizing the scene when the animal moves its head. But does the animal shift its gaze independently of moving its body? Do they saccade around the visual scene? So to look at that, Angie looked at the traces of eye movements relative to movement, to the animal's movement. Down here is the mouse's running speed. So you can see here he's stationary, then he starts running. Then if you look at the eye position traces, you can see that while the animal's stationary, the eyes are also pretty much stationary, except for a very slow drift. Then once the animal starts moving, the eyes begin jumping around, the animal becomes stationary, the eyes stabilize, and so on. So it's clear just from this individual trace here that even when there's a cricket in the arena that the mice might want to be looking at, the eyes themselves are very stable. So they aren't saccading around the scene to try and find the cricket. And in fact, this was true if we look at the distribution of eye speed when the animal is stationary. The eyes are pretty much always completely stationary. Whereas once the animal is moving and running about, there's a wide distribution of eye movements. Okay, so animals aren't sitting, when they're sitting still, they aren't saccading around. All the eye movements occur when the animal starts moving. But what is the pattern of eye movements? What are they doing when their head is moving? So to look at this, Angie looked at the relationship between movement of the head. So this is the horizontal or yaw velocity, how much they're moving their head side to side versus how much their eyes move. And so all the points along this diagonal line correspond to those compensatory eye movements. So if you move the head one way, the eyes have to move the exact opposite way to compensate. And so therefore you get this linear inverse relationship. So I should mention each of these individual time points is a single frame of the movie looking at what happened to that mouse's, how that mouse's eye moved relative to its head. So dark points are all of those compensatory movements. All of these red points are times when the mouse's eye movements weren't compensating for the head movements. They were moving in a different pattern that would not cause, that would not compensate for head movement. So to figure out what these non-compensatory movements are doing, Angie put together the trace of the animal's head position with the trace of the animal's eye position to calculate where the eyes are actually pointing in space. So the direction of gaze is going to be a combination of where is your head plus where is your eye pointed, which we call the gaze direction. So here you can see that in this case, the animal's head was slowly rotating while the eyes were doing this series of slow movements and then jumps, slow movements and jumps. When you add these together to figure out where the eyes were actually pointing in space, you see this very distinct pattern that the eye position will be very stable. And then once you have one of these non-compensatory eye movements, there's a big jump and then another stable period and a big jump. So effectively what this shows is that what the eyes are doing is, in general, as the head is moving, the eyes will compensate up to a point and then they'll jump, create a new gaze location, stabilize that new gaze location, and then if the head keeps moving, jump, and so on. This pattern had actually been described in another species by Michael Land, who called it the saccade and fixate pattern. And in some ways, it's very similar to the way that we saccade and fixate, except in this case, the saccades are driven by head movements as opposed to spontaneous saccades. Okay, so together, these compensatory and non-compensatory movements are creating a stable image, which then jumps as the animal's head moves. But the question we wondered about is, these saccades, when they choose a new location to fix their gaze on, are they maybe then targeting the cricket? So to look at this, Angie looked at both the head position and the gaze position relative to the cricket every time the mouse made a saccade while they were hunting the cricket. And so the head position relative to the cricket is shown here. So you can see that, on average during the pursuit, they're turning towards the cricket. They're always continually updating their trajectory to be aimed towards the cricket. But around the time of the saccade, you can see that right before the saccade, the gaze, the direction of the eyes in the world is stable. So again, they're stabilizing the gaze as the mouse is aiming towards the cricket. And then there's a jump. This is the saccade. But what you can see is the saccade jumps to the same direction as the head. So it's not like the gaze is all of a sudden jumping onto the cricket. That would be jumping down to azimuth 0. It doesn't target the cricket any more precisely than the head. So together what this says is that the orienting of the mouse relative to the visual scene is largely driven by the head. The head is determining where you're looking. And what the the eye movements are doing is stabilizing this and then catching up. I should mention that there have been a couple of recent papers, one from Jason Kerr and one from Matthew Self, showing that mice do tend to keep targets in a particular region of the visual field. But again, and in fact, Jason Kerr showed that this corresponds to a region of higher alpha ganglion cell density, so something that maybe could be considered a phobia. But again, it was always the head that was getting the visual scene into that location as opposed to the eye movements themselves. They aren't saccading to that location. So on the one hand, this was great for us to finally know, again, after years of watching mouse behavior, to finally know where they're looking. But this is also interesting. This pattern of saccade and fixate, like I mentioned, it's similar to what is seen in primates. And a big question is, how does the brain stitch together this series of stable images into a coherent representation of the world? And so now that we know that mice engage this mechanism as well, we can start to go in and look in the brain, into cortex, and figure out how you create this stability across eye movements. Okay, so that's a little bit about what mice can see and how they do it by moving their eyes. Okay, of course, a big question is, what are the neural circuits that are mediating this? And this is also work by Jen Hoy. I should mention she now has her own lab at University of Nevada. So any of you who are interested in this work, I should also check her out. And Jen was immediately attracted to studying the superior colliculus as a potential brain region that might be mediating this. The superior colliculus is a region in the brainstem. It's highly conserved across species, from frog all the way up to primates and humans, and of course mouse. It receives direct visual input from the retina. And there's several reasons we were interested in it for prey capture. One of which is that in other species, such as frogs, fish, and owls, the superior colliculus, or the equivalent in non-mammalians, the optic tectum, had been shown to be involved in prey capture. So it seemed like a very likely candidate. The superior colliculus also has an anatomical organization that's really almost ideally suited for something like prey capture. It's a laminar structure that gets visual input into the superficial layers. It gets multimodal input, auditory and somatosensory, into the middle layers. And then the deep layers are a motor output. And all of these are aligned so they correspond to the same region of visual space. So a given point on the superior colliculus as you go through all corresponds to the same region of visual space, either to a visual stimulus at that location, an auditory stimulus at that location, or to drive a motor output to orient towards that location. So you can imagine, basically be ideally suited to detecting here's the cricket in region of visual space, and then turning towards it and approaching. So its structure, suggested it would be ideally suited for this. We were also interested in using prey capture as a way to understand the superior colliculus. So, for over 100 years, this is a drawing from Ramon y Cajal showing the different cell types. We've known a lot about the structure of the superior colliculus. We know about the receptive field and response properties of neurons. We don't necessarily know how these different cell types get engaged in behavior as the animal is actually interacting with the visual scene, or interacting with the visual world. What do these different cell types do to contribute to visual behavior? So we were interested in two in particular. One is, both of which actually were described by Ramon y Cajal in that illustration I showed a minute ago. One of which was the wide field vertical neurons, and the other is narrow field vertical neurons. And I should mention that both of these two were identified as Cris lines by Gabe Murphy a few years ago, who performed a very nice characterization of their anatomy and function. These wide field vertical neurons have very large dendritic arbors. So you can imagine they'd be good for detecting, integrating information over a large region of space. So to kind of detecting whether something's there. But with that large dendritic arbor and corresponding receptive field, they're not going to be able to very well localize a stimulus within that region. They also provide their output to pulvinar, which, as we came up in the last questions, can distribute visual information to multiple areas across cortex. So the combination of its large receptive fields and its projections up to other cortical areas suggested to us that it might be involved in detecting whether the cricket's there, alerting the rest of the brain, and initiating hunting. On the other hand, these narrow field vertical cells have much smaller dendritic arbors. So they're kind of ideally organized to be able to detect exactly where a visual stimulus is. They also provide their output to the deep superior colliculus, which I mentioned is the motor output. So they're also well suited to drive an output to orient you towards that location. So this suggested to us that they might have a role in determining exactly where the cricket is, and then turning towards and approaching that location. So again, our hypothesis that the wide field cells are basically like large field antennas that can say where something's out there, but not where. So involved in detection, and the narrow field cells are much more tightly localized and could drive direct approach. So to test this, Jen used a chemogenetic approach using the Cris lines that Gabe Murphy had developed that gives us access to these wide field cells, the narrow field cells. And then as a comparison, we studied a population of PV cells, the PV Cris line. And the superior colliculus, this is actually a mix of excitatory and inhibitory neurons. For those in the cortex, we think of them as always inhibitory. But the reason we chose this as a comparison is that other groups had shown that this population was involved in the fear response. So if you have a looming stimulus overhead or a flash stimulus, an animal will freeze, and they had shown that these PV cells mediated that. And so we thought that'd be a good comparison to cells we thought might be involved in prey capture. So Jen used chemogenetics dreads to inactivate these cells. I'm happy to talk about some of the controls that we did to test for the specificity. But the effect that she got when she shut down these different cell types was quite dramatic. So the control animals, catch the cricket within about 5 to 20 seconds. The PV cells, when we inactivated the PV cells, we saw very little impact on the behavior, kind of consistent with the fact that they are detecting looming or dark stimuli, evoking fear responses, as opposed to detecting something like a cricket that you would want to approach. Whereas when we shut down both the wide field and narrow field cells, we saw a deficit in the time to be able to capture the cricket, which was greater for the narrow field cells. What was most informative, though, is when we went in and looked at the specific pattern of deficit when we shut down these two different cell types. So on the left, you'll see the effect when we shut down the wide field cells. You can see the cricket's there. The second here, the mouse, kind of almost looks directly at the cricket's location. So by now, the mouse would have normally detected the cricket's there and begun pursuing it. Once the mouse does detect the cricket, realizes it's there, particularly at close range, it's still able to pursue the cricket, but then it loses it. Almost, can't find it again. Once it finds it again, it can go straight towards its location. So it really seems like it can't figure out when the cricket, it has a much harder time determining when the cricket is there, but once it does, it's able to make those approaches. On the other hand, when we shut down the narrow field cells, they still have a hard time figuring out the cricket's there. Once they detect it, though, they aren't able to make an approach to it. They take this wandering path, they stop. In a second here, the mouse actually freezes right in front of the cricket. It's like it's not able to generate that motor output towards the location of the cricket. So in order to quantify these, Jen calculated a couple of different parameters, actually a number of statistics that kind of fell into three categories. One is the ability to detect the cricket. I'm just showing one of these here. This is the time that it takes to make the first approach towards the cricket's location. So basically to start hunting. When we shut down both the wide field and narrow field cells, the animals had deficits in that. With the wide field, we think it's more about the ability to detect that the cricket's actually there, because it turns out they can do it relatively well once the cricket's close up. With the narrow field, we think is that they can't make that turn that initiates the first approach. The second was the ability to target the cricket. And here we measured this by the probability of intercept. So once they started chasing the cricket, do they actually get to the cricket's location? And so typically mice, in any of the, when either in wild type or when the other cell types are shut down, once they start approaching the cricket, they always get to it. Sometimes the cricket will escape, but they're always able to get to the cricket's location. Whereas when we shut down the narrow field cells, they weren't able to target because they were taking these wandering paths or they would stop. And then the final measure was pursuit continuity. And we measured this in terms of the fraction of time that the mouse was immobile in the vicinity of the cricket. So as I mentioned, in a wild type mouse, once they're near the cricket, they're always going to jump and try and catch it. When we shut down the narrow field cells, they actually spent a significant fraction of their time immobile, not able to finally complete the pursuit, as if they couldn't, they were lacking the ability to see where the cricket is and drive towards that location. So put together, this suggested to us, that the wide field cells were affecting the ability to detect and kind of start pursuit, whereas the narrow field cells were involved in that precise targeting and ability to get to the location of the cricket. We went back and did a little bit of electrophysiology just to measure the receptive fields and see if they matched up with this. As you mentioned, we had to go back to a head-fixed recording, present visual stimuli. I'll come back to address that concern a little bit towards the end of the talk. But we were able to show that the receptive field properties matched up with these roles in either detection or localization. And the wide field cells had very large receptive fields, responded to both light and dark stimuli. The narrow fields had much smaller receptive fields, ideal for localizing the cricket's location. Whereas these PV cells, the ones that had no effect on prey capture and have been shown to be involved in fear responses, responded to much larger stimuli and typically responded to dark stimuli, just like you would expect for an overhead fear stimulus. Okay, so that's a few of the things that we've learned from studying prey capture. I first showed you, what are the parameters of mouse vision, how far away, how large can they see. I told you about how we use this to figure out where and how do mice direct their gaze. They're largely directing where they're looking based on where their head is, and then the eyes are creating a series of stable images interspersed by saccades. And then finally, I told you about how we'd use prey capture to try and define the role, to figure out the role of these defined cell types, ones that have gone back to Romanika Hall, and figure out not just what are the response properties, but when do these cell types actually get engaged in particular visual behaviors. So since this is a symposium on cortex, I thought I would now just show you a little bit of new preliminary data addressing the question of, well, what's the computational role of cortex in this? Is it all just superior colliculus? And one thing I should note is that the paradigm I've been showing up till now was really the simplest possible visual task you can imagine. There's a dark cricket on a white background. nothing else in the arena. So all the mouse has to do is find the dark object and go towards that. And that's really what superior colliculus is ideally suited for. It's basically, kind of a feature detector, even going back to the old paper where the frog's eye tells the frog's brain. And so this, led us to hypothesize that what superior colliculus does in prey capture is it performs kind of low-level image processing. It can do a contrast detection. There's a dark object there, and it can localize it and drive that approach behavior. However, of course, there's also the pathway through cortex. So the eye via LGN projects up to visual cortex, and visual cortex also sends projections down to superior colliculus that could help guide this orienting approach, the motor output that's generated by superior colliculus. But the question is, when would cortex be involved in this? What can cortex do? What computations does cortex perform that superior colliculus cannot? And so we hypothesize that cortex might get involved when, rather than just detecting a simple dark spot, the mouse actually has to do something more complicated, more complex image processing. For example, if the cricket is camouflaged, or if it's on a complicated background, or for example, if you have to discriminate whether this is a cricket that I want to approach or some other object that I either don't care about or that I want to avoid. And so we hypothesized that visual cortex performs nonlinear image processing, extracting information that's not explicitly represented in the pixel-wise image of the visual scene. So things like image segmentation, detecting a cricket on complex backgrounds, or discrimination, distinguishing a cricket from other objects. So to test this, we measured the animal's ability to do prey capture in different environments and with and without inactivation of cortex. So this shows the 3 different environments that we used. One is our original white plain arena. The next was an arena that had black and white camouflage along the walls. So that mouse now has to segregate out the cricket relative to that background. And then finally, that same background, but now with black and white objects. It can both serve as distractors, but also as occluders that the mouse would have to look behind in order to find the cricket. So this shows the results quantified in terms of the time that it takes the mouse to capture the cricket. If you just look at the times when the laser is off, you can see that as we make the environment more complex, it takes slightly longer for the mouse to be able to do that, so it becomes a harder task. Then you can see that once we shut down cortex, we start to have a very profound effect on the mouse's ability to do it. And such that in that most complex environment, it's really quite difficult for the mouse to be able to catch crickets. So this suggests to us that, that hypothesis that the visual cortex can basically play an additional role in prey capture in these complex environments, basically in cases where you have to do something besides just contrast detection, where you have to draw nonlinear operations like image segmentation or discrimination, dealing with occluders and so on. So on one hand, this is actually, it's kind of a nice, provides some new understanding of the relative role of cortex and superior colliculus, what the computational role is for each of those, but also now allows us to start going in and saying, now we can study things like image segmentation or object recognition, but in this natural task, as opposed to, for example, with a 2AFC. Okay, that's the end of the section on prey capture. In the last few minutes, I'd like to tell you about a couple of new approaches that we've been working on to study vision and movement together. So the first is a project that was developed by a remarkable postdoc in the lab, Phil Parker, who was interested in studying depth perception and particularly through motion parallax. So motion parallax is a phenomenon that as you move your head side to side or back and forth in the world, objects that are close up are going to appear to move a lot, and objects that are far away are going to move much less. And this is actually a very powerful cue for 3-dimensional or for depth. For example, if you close one eye and then move side to side, all of a sudden you'll get a pop out of 3-dimensional perception, the 3-dimensional nature of the world. And in addition to just kind of being a little interesting novelty, this is actually a very strong cue for depth. For example, as you're navigating through the environment, if walls are closer, they're going to generate more parallax. We tend to steer away from that. So Phil was interested in seeing whether we could study this in an ethological context. And he actually drew on some experiments that had been done back in the 80s by Mel Goodale, who showed that Mongolian gerbils can jump across gaps and that they use this motion parallax cue to be able to do that by bobbing their head up and down to generate motion parallax. So Phil set out to see if our mice could also accurately judge depth at jumping across a gap. And so he trained mice to jump across gaps. Mostly he just had to motivate them by putting a little bit of food on the other side of the gap. But mice can jump across both short gaps as well as these much larger gaps. So again, kind of like the crickets, I was pretty impressed that our mice are capable of these physical feats. But more importantly was the fact that, first of all, they have to use vision to do this. We make the gap far enough that they can't use their whiskers. You can't smell how far it is across a gap. But also, Phil went on to show that they do this accurately and that they are likely using motion parallax to do it. So he quantified the behavior, again, using, as everybody does now, deep lab cut to track the animal's kinematics as they're jumping across the gaps. They do this quite accurately. Most of their trials, they're successful, and they land basically with their nose just on the far side of the platform. They occasionally fail by jumping too long or coming up short, but overall, they're quite accurate. This plot here shows the distance across the gap versus how far they jumped. I kind of think of this as a psychometric curve, but for jumping. And you can see that they jump quite accurately, but coming up sometimes a little bit short for the furthest distances. They also do this, in general, they're quite successful. Most of the time, they do land on the platform. They start to fail when the platform gets too far away. But Phil went on to do an important test, which is to cover or to close one eye of the mouse. So they only have monocular vision. And he found that they're able to do this task almost exactly as precisely when one eye is closed as when both eyes are open. So this shows that they don't need to use stereo vision to be able to do this. They aren't necessarily using binocular stereopsis, but they most likely are using the motion cues that are present when one eye that you can use with monocular vision. So he went on to see, what are the movements that the mouse is making, and do any of these predict the animal's performance on the task. To do this, we teamed up with Luca Mazzucato, who's a theorist here at University of Oregon. He implemented an autoaggressive hidden Markov model. Basically, what this does is it breaks up the continuous dynamics of the behavior into individual motifs, and then says what's the probability of going from one of these motifs to the next. Kind of very similar to the MoSeq approach that was developed by Bob Datta's lab. So we get different movements, the transitions between them, which kind of lets us see the sequence of events that occur before a jump. This can be seen here. This is showing each of these kind of movement trajectories. Blue is the start of the movement, red is the end, and kind of the sequence through the jump. What Phil found is that one of these particular movements, towards the beginning, the mice make this upward head movement. And they make this upward head movement, and that this particular movement was both more frequent when the animal was successful. So the dark line shows success, gray shows failure. So these movements were more frequent, particularly at short distances when they were successful, and they were also larger amplitude. Of course, a larger amplitude movement would give you more precise information about the motion parallax. So of course, any movement will generate motion parallax, but it suggests that this one might be particularly important in generating that parallax. And so then finally, Phil went on to test whether visual cortex is necessary for this. So he covered one eye, so we're forcing them to use motion signals. And then I should also mention, I'm not talking about here, he did a series of controls to rule out the possibility that they might be using retinal image size. So he used 3 different sizes of platform. Retinal image size is, if an object's further away, it's going to appear smaller. That would be an obvious cue, but he used different sizes of platforms to eliminate that possibility. So he covered one eye, shut down visual cortex bilaterally. When we use control animals that aren't expressing PV channel rhodopsin, so we just have the light only control, they jump just as accurately. Once we suppress V1 activity, their performance is dramatically impaired. So this demonstrates that they need visual cortex to be able to judge depth in this task, or that visual cortex makes a large contribution to their ability to judge depth in this task. So for us, this task is quite exciting. Not only is kind of a new thing that the mice use their vision for, but in particular the fact that this is something that combines movement and vision for a very specific computation. Unlike in the beginning, I mentioned that this gain change, and we don't necessarily know what it's trying to achieve. Here we know what the computation is. It's combining self motion with retinal motion to calculate distance. And I should mention, Phil is actually currently on the job market. So hopefully when he sets up his own lab, he'll be going in to look at those circuit mechanisms that are bringing together movement and visual information in V1. So of course, you probably recognize by now, there's a big challenge in all of this, the freely moving behavior, which is the ability to record visual responses. How do we perform visual physiology in a freely moving mouse? How to measure visual responses? And that's because the key thing that we do in visual physiology is there's a stimulus at this location in space. What's the neural response to that? And once an animal is freely moving through their environment, we no longer have any control over that visual stimulus the same way that we do when we're presenting it on a monitor to a head-fixed mouse. Instead, the mouse is in control. They're choosing what they're looking at. And so we need to know not only what are they looking at, what's the scene in front of them, but where are their eyes pointed within that scene. So to address this, Phil, again, has been leading the effort to set up a system to allow us to do visual physiology in freely moving mice. This combines a drivable silicon probe, a chronic implant that we put into visual cortex, with now 2 head-mounted cameras, one of which is actually looking out at the visual scene so that we can see what's in front of the mouse, and one that's looking back at the eye so that we can correct for where the mouse's eye is pointed within that scene. And of course, we also have an overhead camera to see where the mouse is moving through its environment. So this is what it looks like when we put it all together. You may or may not be able to hear the spikes. I'm not sure if I turned on the sound. But you can see here, we're recording about 30 units simultaneously. We can see where the eye is pointed. We can see the visual scene that the animal is encountering. We can get measures of the eye and the head movements. You'll see in a second here, once the animal sits still, the firing really drops off. But so now what we're doing is we can actually record the activity. We know what the visual scene is in front of the mouse. We know where their eye is pointed, and we have the activity of a large population of units. So, the first instinct might be to do the same spike triggered average that I mentioned at the very beginning. It turns out that that approach doesn't work here because, natural scenes, and particularly natural scenes as an animal's moving through it, are much more complicated. There's large correlations in the stimulus. Also, certain stimuli are we aren't controlling what the mouse chooses to look at. So some stimuli are overrepresented, others are underrepresented. But it turns out we were able to take a different approach. We implemented a generalized linear model, or GLM. And we used this to fit not only the response to the visual stimulus, but also to head and eye movements. For those who are interested, this is just a simple linear, nonlinear Gaussian model, but with a strong smoothness prior on the visual input. So you can see for this unit, we mapped out a receptive field that looks much like we might have gotten from a head-fixed mouse. It's a dark spot with a light edge. We're also able to get the kernels for the eye movement response. In this case, the cell is suppressed right around the time of a saccade and has a strong response afterwards, as well as a response to head movements. So we're just starting to dig into all this data. The analysis, as you can tell, is taking quite a bit of work. But what we're excited about is finally the ability to know, what's the visual response of a neuron as it's moving through the world, and how that relates to these head and eye movement signals. We're also particularly excited about looking, this is just looking at kind of the two-dimensional structure of the image. What we really want to do is be able to measure the response to stimuli, to feature the visual scene that aren't present in a two-dimensional stimulus in the monitor. Things that are inherently part of your three-dimensional movement through the world. So things like motion parallax, object continuity, occlusion, and so on. And then finally, to be able to apply this approach to those behaviors I described earlier, for instance, to be able to measure the responses in visual cortex as a mouse is hunting a cricket in a complex visual scene. So just to summarize this last little bit, our new approaches, on the one hand, we're trying to take features, visual computations that we might have studied in a 2AFC task, like distance estimation, but convert them into an ethological behavior, and in particular, something that combines movement and vision together, and therefore could only be studied in a freely moving context. And then finally, to take the same types of measurements of neural responses and receptive fields that we would have done previously with a head-fixed animal looking at a computer monitor, and now be able to measure these visual responses as the animal is moving through and interacting with a three-dimensional natural environment. So with that, I'd just like to thank all of the people in the lab, past and present, who have contributed to this. I tried to mention the people who contributed to specific things as I went along, and then our collaborators and funding sources, and happy to take some questions.